Good afternoon.
All right, that's it. All right,
Okay.
welcome. All right, yo. Welcome to the first episode of the Might of Chondrea, a sort of podcast-esque study thingy that we're doing because we want to, and it'll help people, maybe. Yeah. All right,
Studying anyway, so join us, I suppose.
that's true. Yeah. Uh, I mean, I'm sure introductions are not necessary, but and so we won't do that.
Yeah, if you're here, you know who we are.
Yeah, if you're here, you know who we are, and if you don't, well, why are you here?
Good
Fair point.
afternoon.
All right, he did it again.
Okay.
Yeah, it's time to get started. This time we're gonna be reviewing Unit 5, Animal Physiology, but again, which basically nerves and muscles.
Four.
Everyone's
Unit
favorite.
4.
Yeah.
Wait, with
Oh, is that
5?
4? That's
Oh, I don't
Oh,
know. The
five.
wait, the doc says 5, but if you scroll down, it says 4. Whatever.
whatever. Nerves and muscles. That's what we're here to do. Every 30 minutes, we'll be taking a little break. Healthy study habits are being promoted here.
Yay.
All right, so I don't know. Let's get started. So,
You
obvious.
wanna show me your notes? So I'm not.
All right, cool. So, starting off, what is a nerve? The nervous system is divided into the peripheral and the central nervous system. The central nervous system is composed of the brain and the spinal cord, while the peripheral nervous system has two systems within it, the autonomic or autonomic nervous system and the somatic nervous system. The somatic nervous system is what we mostly focus on here. So it's the sensory and the motor nervous system. Allows you to sense things and move. That's what that means. And the autonomic nervous system, which involves the parasympathetic and sympathetic. So these are automatic responses that your body normally does, like keeping your heart pumping or shivering when you're cold, sweating, things like that. So the central nervous system performs coordinating tasks. That's what the brain and the spinal cord do. They propagate nerve signals throughout the body and sort of regulate everything, and that's cool and good. Somatic means voluntary, and autonomic obviously means involuntary, which I was explained earlier. If you don't understand that by now, then okay. Moving on. So, Justin, what are the differences between the central nervous system and the peripheral nervous system?
Well, that's a very good question. Let me answer it.
Yes, that is why I asked you.
I will now answer the
Oh my
question.
god. Oh my god.
Just go.
Just go.
Okay, so the parasympathetic nerves. Wow, it consists of nerves. It connects the brain and spinal cord to the rest of the body.
All right, yeah. But
They
the difference there
constrict
is it
pupils.
consists of just nerves, right? Like there's the
Yeah, yeah,
brain's
yeah,
not
yeah.
involved, the spinal cord's not involved. It's only the nerves. That's the
Right.
important takeaway. Sorry, continue.
Yeah, yeah. It's used to constrict pupils, stimulate saliva, slow down your heartbeat, constrict airways, stimulate activity of the stomach, stimulate the gallbladder,
So
stimulate the activity of intestines, and
like
contract the
all of
bladder.
the automatic stuff then, right?
Yeah, yeah, yeah.
Okay.
They can also do the reverse, like dilating them, inhibit salivation, and so on. The central nervous system is composed of the brain and the spinal cord, uh, and it's surrounded by bones and vertebrae. There's also fluid, and there's a tissue that insulates the brain and the spinal cord. And the fluid's called cerebrospinal fluid.
Yeah, fair enough. So you mentioned earlier about constricting pupils, and then the sympathetic response is the opposite, which would be dilate the pupils. So what's the difference between sympathetic and parasympathetic?
Parasympathetic is when you're in this sort of state of rest and relaxation, and the sympathetic nervous system responds to stress or the fight or flight response.
There we
Okay.
go.
All right. So I guess what's next is neuron structure, right?
Mm-hmm.
Cool. Uh,
Yep.
okay, I guess I'll read it. So the glial cells, they're non-conducting cells that are important for structural support and metabolism of the nerve cells. Uh, there's also Schwann cells. Is that what they're also called? Yeah. So the
Yeah.
Schwann cell is sort of like around the axon of the neuron. It's sort of like a protective layer. It has a myelin sheath, and this just aids in
The
the
Schwann cells make the myelin sheath.
yeah, they make the myelin sheath. Yeah. So they just aid in sort of the conduction of the action potential. We'll get into that, obviously. So first, let's just look at the structure of the neuron. What is a neuron? That's not a rhetorical question. Answer. What's a neuron?
It's a nerve cell.
Right.
Mm-hmm.
What do they
And
do?
what do you
Conducts
want?
action potentials or electrical impulses.
Right. And
There's a lot of them,
a lot of them.
billions.
Billions, even. Wow. All right.
Yeah,
Cool.
it's crazy.
All right. So let's look at the parts of them. What is a dendrite?
It receives information or the action potential. It conducts nerve impulses towards the cell body.
Right. And so a dendrite, the word itself, dendrite, means sort of like a branch, and you can see that in the structure. If you look at a diagram of a neuron, there's branch-like protrusions that sort of are attached to the cell body. So that's how you remember dendrites. They're branches. That's what the word means. All right. Moving on. What's an axon?
It is an extension of the nerve cell or the neuron cytoplasm, and it projects nerve impulses from the body of the cell.
Okay. We talked about this before, the myelin sheath. So many, but not all, axons are covered in a white coat of a fatty protein called myelin. So they're myelinated, which acts as a form of insulation for the neurons. And so this is form what's called the white matter of, I guess, the neuron. So moving on to the Schwann cells, we touched on this before. It's a type of glial cell. They create the myelin sheath, so they're situated on top of the axon of each neuron. The nodes of Ranvier, I guess Ranvier,
Renvier.
Ranvier.
I don't know. That's what
We're in
Well, I mean,
we were
Canada.
taught.
we don't need to know how to say it. We just need to know how to write it. So they're areas between the sections of the myelin sheath. So I mean, they're typically just called nodes or like inter.
It's a
myelin
node is
nodes,
just
but
where things
It's
intersect.
where there's no myelinated
yeah,
sheath.
there's
Yeah,
basically, if
Like
you
in
imagine
between
the
them.
myelin
just in
sheath like
between.
this covering, like a sheath of a sword.
Sausage links.
Sausage links. That's true. They do kind of look
What?
like that. They're sort of the sections in between each portion of the
Or
myelin
dog.
sheath. So that,
What? I'll send you a picture.
all right. So there's also a neurolemma, a delicate membrane that surrounds the axon of some nerve cells. It's believed to promote the regeneration of damaged axons, but if you damage the nerve cell body, that's non-recoverable. That's pretty permanent. There are how many types of neurons? There's like, we have three written
3.
down here. So multipolar, motor, and sensory. Multipolar neurons, or interneurons rather, inter as in between, they conduct signals throughout the nervous system, so they're in between the more important ones. They just pass the message along, so to speak. Motor neurons, motor, obviously the word means movement, so they have a cell body at the end of the neuron, and they conduct information away from the central nervous system towards, the muscles or the organs, wherever might need to have movement. Movement needs to
Means
happen.
it's efferent,
Efferent. That's
conducting
a word.
away.
Conducting away. How do you remember that?
Sensory neurons are the opposite
Sensory
of
neurons are
motor
opposite.
neurons.
Yep, they
Yeah,
are
basically
afferent.
you go from a receptor cell
Whoa.
to send information to the central nervous system. So yeah, afferent, afferent, afferent.
Yeah,
Afferent.
efferent and
Yeah.
afferent. So E is like, so efferent, it conducts away, so it's exiting, I guess.
E-way.
E for
Exit.
exiting. E for exiting, and
E-way.
A for
Don't say away. Oh my god, that's so
A
confusing.
for arriving. Yeah, E for exiting, A for arriving. Efferent, afferent. Also, sensory neurons have the cell body in the middle of them. All right. So I guess that's the structure of neurons. That's the simple part. Now we move on to the actual sort of ion electric mechanics of it all. Everyone's favorite, I know. So let's begin with the resting potential. So
Okay.
the neuron structure really like contributes to this, obviously, because it's myelinated. They have Schwann cells on the axon, which create and form the myelin sheath. Some neurons are unmyelinated. There's no myelin sheath. I mean, obviously, we'll see the difference and why it's important later. But for now, just know that there's two types. The action potential. So basically, information is sent through the nervous system using an electrical impulse called an action potential. Potential, so it's like an electric potential energy. An action because, it's doing something. It's acting. And these are not formed through necessarily the movement of electrons, but rather ions and an electric gradient formed in that respect.
Electrochemical
Electrochemical, that's what it's called? Right. Yep.
gradient. Pretty sure.
Yeah.
Oh, yeah. You're right.
I am.
All right, so back to what we were talking about initially, the resting potential. So when neurons are at rest, the inside of the axon is negative relative to the outside. The potential difference in charge is called the resting potential because it is resting and there's potential energy. This potential difference can be measured using a voltmeter. I mean, because there's an electrical difference, so obviously it's detectable. The action potential reverses the resting potential, so the inside temporarily becomes positive to the outside, whereas in resting, the inside is negative relative to the outside. And this obviously can also be measured with a voltmeter. So how is the resting potential established? Does someone want to take that? I don't know, go get some water or something.
Yeah, so the main thing that creates this resting potential is your sodium potassium pump.
Hey.
So the cell will use ATP to move sodium and potassium in unequal proportion, so they're not going to be moved at the same rate. And then this is what causes the difference in charge. So you'll move the proportions, or the ratio is 3 sodium out and 2 potassium in. So what you end up having is the inside becoming more positive or more negative? More negative.
More negative.
Or negative.
Yeah. The inside is more negative than the outside.
And what's the I guess you would call it the equilibrium point? Like what's the difference of charge that the body tries to maintain?
Negative
I mean, if you're
70.
moving 3 out, 2 in, it'd just be negative 1.
Negative
So
70 millivolts.
Negative 70 millivolts, yeah.
Yeah, so the pump basically sends out 3 sodium and brings in 2 potassium ions. So it's getting a little bit more negative each time because 3 positive charges are going out, 2 are entering. And so eventually it wants to maintain that sort of negative 70 millivolt resting potential. Using the terminology. Wow, incredible. Yep, okay, so if you look at a diagram sort of how this whole thing happens, we need a charge difference. So there's countertransport, as we've already said. Did you guys talk about leak channels?
Not yet.
All right, so potassium
I
and
was trying to
sodium.
segue into it.
Right, well, I mean, we've successfully done that. So
Hey,
there's potassium and sodium leak channels. What's a leak channel?
Justin, go ahead.
it's just where the sodium or potassium ions can just diffuse right back through to maintain equilibrium because you made the charge difference, right, to do the thing. And then now the ions got to go back to equilibrium. So they just diffuse through the leak channel.
To maintain the negative 70.
Yeah, to maintain the negative 70.
All right.
Okay.
Okay, so neuron action unfinished. I don't know why
we didn't finish it.
called it that. Yeah, I guess this part's unfinished for us. However, I mean, we'll ad-lib something. We'll figure it out.
See if I can figure it out.
Sure. So the motor system is like the central nervous system initiates the thought that you should move. So if I were to lift my arm here, obviously I think about lifting my arm, and then I move my arm, and while I'm doing that, the brain obviously sends a signal. For sensory neuron action, the it makes use of the peripheral nervous system. There's usually some type of receptor, like a taste receptor, smell, olfactory receptor. Is that what it's called?
Yeah.
Yeah.
Yeah. Or like did I say, yeah, pain receptor, etc. They sort of get some type of stimulus, some input, and they send the signal. So how do we sort of regulate this? If you could feel, every slightest change in the air current around you, it's probably way too much information. It's not useful evolutionarily wise. So how do we regulate this? What's the solution?
It's a good question.
Voltage gated channels.
Voltage gated channels. As you can tell, they are gated by the voltage. They only open under a certain situation, and that is when the membrane potential or the resting potential reaches a certain point. Call it a threshold. It's just a threshold. Yep, cool. So a concentration
Yeah, so
gradient is made between sodium and potassium. You can continue. I cut you off there.
yeah. Oh, it's fine. I was just going to say, in the resting state, your voltage gated channels are going to be closed entirely. What did we say? What exactly the threshold is? I don't know if it's like an exact thing.
It is
Around
an
negative
exact thing, but
50?
it's yeah, it's
I'm
negative
scrolling down.
50-ish.
Around negative 50, I think.
Yeah. All right. Well, we'll get there. So first of all, let's back on the topic of voltage gated. Um, so there's an increase in the membrane potential as the concentration changes. So essentially, once the stimulus is sort of achieved, the inside begins to gain more positive charge through the movement of sodium in, based on whatever stimulus is there. I think there's a system in place that allows for that to happen. I don't know if do we have to know that in particular? Like what causes the change in voltage? Or is it just stimulus causes a change in voltage and that's it?
I think we just need to know, like, stimulus.
Right, okay, cool. So
I just know it's a sensory
so
thing.
stimulus causes a change in the voltage. The inside starts to get more positive because sodium is coming in. And so if the stimulus is stopped there, then it'll just rebalance itself out. Nothing will happen. However, if this is allowed to continue and get stronger, to reach what is called the threshold potential, as we said before, there is a sudden fast spike in the membrane potential, because once that threshold potential is reached, then the floodgates are open, so to speak. And so a bunch of sodium channels start opening up, and it's just completely flooded, and so a spike in voltage occurs. It goes from negative 70 and it goes all the way to like positive 25, 40. It's just something above zero, pretty much.
Yeah.
And
Yeah, so there's potassium channels and sodium channels, but once you hit that negative 50 threshold, the first step would be opening the sodium channel. So the sodium that has been expelled as like during the resting potential that caused that like inequality, that's going to start rushing in.
Yep.
And that is your depolarizing phase.
Cool. So this electric pulse is then generated and propagated throughout the nerves. It's called the action potential. As we said, it reverses the resting potential because of all the sodium flooding into the cell. The axon rather, the inside becomes positive relative to the outside. Great. So essentially, there's sort of 4 phases in terms of like how an action potential gets generated. So first we have the resting state, obviously, where where everything is, as we've said, it's normal, it's calm, it's chill, all of the channels are closed, not getting any reception. Okay, all right, bad joke. And then the depolarizing phase, the stimulus has been achieved, or you've thought about moving your arm or your leg or something, and so the gates are open, the sodium gates, which allow the charge to flood into the cell. And so the membrane potential goes from negative 50, which was the threshold, I guess, all the way to something positive like 25, 30, somewhere around there. So that's the sudden spike. So now it's depolarizing because it's, going the other way.
Yeah. So the inside of your cell will become negative as a result.
Positive. The inside's positive.
Right? Look at it.
That sodium is coming in. The positive ions are coming in. The inside becomes positive.
Lol.
Lol. And then the repolarizing phase, in which the sodium channel closes on the inside. So sodium is now trapped inside of the axon, and then the potassium gate opens to release potassium to the outside. And so now the positive charge is exiting the cell, so it's slowly becoming more and more negative. Again, this happens so quickly and so, I guess, effectively that you undershoot, or I guess you go past what you're trying to get, which is, as we recall, the resting potential of
negative 70.
Negative 70. So we go a little bit beneath that. So like negative 80, 75, something around there. And then the sort of regulating system kicks into place. Pumps are now reactive and sort of, get it back to what it's supposed to be. Now we're back at step 1, the resting state.
We can also say that in the depolarizing phase, when Na is in, potassium is prevented from going out, and the opposite happens in repolarizing. There's an inactivation gate preventing.
The back door closes.
Yeah.
Right. All right. So
Therefore.
the action potential only really moves in one direction, because part of the axon is still in the refractory phase, so it is unable to start a new action potential. And so essentially, consider the following, right? Once the signal starts in one spot, that spot becomes positive, and then it starts resetting. Okay, so if you imagine three different sort of parts of the axon, 3 different nodes. The first one, let's say the one on the very left, so there's nothing to the left of it, it's on the very left, it becomes positive, and then it just starts to reset. So the positivity of this part then transfers to the second one because there's like an overwhelming increase in the voltage. So the second one now has also reached that potential, the spike, as a result of the positivity of the first node. So this one goes into that whole thing, becoming crazy positive with the spike. And so when the charge is here, it wants to spread to both the left and the right. But if you remember, the left one is still in the resetting phase. So it is unable to start a new action potential. So the action potential can only move to the right. And this is how the nerve signal, that impulse, that electrical potential travels in one direction throughout the neuron, rather than just being sort of an unorganized, every omnidirectional mess. It only moves in one direction because of the structure. And, it's really cool. There's kind of a cooldown on it, right? It can't fire immediately. And that result pushes the impulse only in one direction. Cool. All right.
Okay.
So that's how one nerve works. But obviously we have how many? Billions. Which is greater, billions or 1?
I don't know. Justin, do you know?
Is
I was drinking
Okay, he wasn't
water.
listening.
I was
Billions.
drinking water.
Is
Billions,
a billion greater
billions.
than 1? Yeah, billions is
Yes.
greater than 1, and we have
Yeah.
billions. So how do nerves work together?
It's something called action potential propagation.
And what does it
And that is
propagate
how
across? What is it called?
the
When
axon.
when 2 neurons meet. It's a synapse. Come on, guys. All right. Well, basically, once the action potential reaches the end.
That's the place.
Yeah. All right. Well, that was whatever.
Maybe I misheard.
Maybe I misspoke. Either way, basically, once the nerve signal, the electric potential, reaches the end of one neuron, it has to move to the next. And this happens across a synapse. So what's the difference between synapses in motor neurons, interneurons, and sensory neurons?
Huh?
All right. Remember before when we said somewhere, um, what did we say? They were efferent and afferent because there's structural differences between different types of neurons. The three motor neurons, interneurons, and sensory neurons. So basically, a motor neurons start off in the central nervous system and they can connect to effectors in the peripheral nervous system. The interneurons are just connections between neurons, and sensory neurons have receptors that connect to those neurons, then transmit the signal back to the central nervous system. It's about the direction and sort of how they work with each other. Those are different synapses.
Did you answer the question?
I answered my own question. So, yeah, I did.
But you said what was
Did
the
you?
difference between the motor neurons, interneurons, sensory neurons, and the synapse?
I
Because
didn't
like those
say
are
any
neurons.
synapse.
Oh, okay. Then that's where I
Oh,
misunderstood.
Right.
The synapse is a spot
The synapse
in
is a
between
place
the
which
an
in between the
region
neurons.
area.
presynaptic
It's
and
a
the
region.
postsynaptic neuron.
Yeah.
Yeah.
Okay. So what happens to a signal depends on where it comes from and where it goes. So neurons connect to each other at a place called a
Synapse.
synapse.
Synaptic cleft.
Don't all come at once now, guys. All right. So there's sort of broadly two different types. There's the electrical and the chemical one. And so electrical synapses, I mean, are pretty standard. It's just directly or close enough that the electrical signal can just pass in between them. Yeah, pretty much, right?
Yes.
Yeah.
You're going to focus on the chemical ones, though.
Because the chemical one is the more complicated one. So, of course, that's what we have to look at.
Yeah.
That's what we're getting tested on, because of course.
Yeah, so chemical synapses. Well, I've said chemicals. What are the chemicals? What is the broad name for them?
Starts with neuro, ends with
Ends with transmitters.
But what is it, Brian?
Neurotransmitters.
Neurotransmitters. Neuro as in brain or nervous system, and transmitters as in conveying a message. So they transmit messages in, from, or through the brain.
Crazy.
What are some
No.
examples of neurotransmitters?
Acetylcholine
Serotonin.
and Brian, go.
Dopamine.
There we go.
Yeah,
3 for 3.
We're insane.
All right, so I
What's
think
the first
that's a
thing
good
that
place to
happens?
take a break. Actually, we've hit
Oh,
just
okay.
about the
We have
30-minute
hit
mark.
the half-hour mark.
Wow. All right. Congratulations. You
Yeah,
have
yeah,
been
we have.
studying, or at least listening to people study for 30 minutes. Incredible.
Yeah.
Up until this point, I guess we've looked at the structure of a nerve, the overview of the nervous system as a whole, the neurons, and sort of what composes them. We've looked at the resting potential and the action potential and how they are created, propagated, and the system that does it. And we've just briefly begun to look at what a synapse is and that there are different types. I mean, yeah, so I guess we'll take like a 5-minute-ish break. That time has
Thank
come.
you, Hank Green.
Everyone's tired. That was my best Hank Green impression. Wasn't really doing a voice, just the intonation, but that's okay. All right, so I don't know, man. What do we talk about? Interesting. Okay.
No?
Talk about What was that?
Hmm?
No, I
I'm
didn't.
just
hear
going to say, we are getting forced to go back to school. Seems very relevant.
That's true. Yeah, I mean.
Still don't know what cohort I'm in.
I mean, it says if
you're
you look on.
Yeah, it says. Go to
But
teach.
like, is it? I don't know what the other. I have my numbers, but I don't know what the other
like-
half would be.
It's either 0 or 8.
I think, yeah.
I don't have zeros or eights.
Well, if it's 0 to 50, then or 0 to 49, I'm not too sure. Actually, no, it's 0 to 50. You're in cohort A, then 51 to 100.
Why wouldn't they just do 1 and 2? Like, I don't understand the purpose of this.
it's
Maybe it's
I
something
don't
for
know.
the
It's
super
some
secret admin.
For A or
system
B?
thing. I
No?
don't know. I think it's just how the system works with the numbers and such. They can't really. Anyways, I feel like that's too specific. You know, you're gonna get
Do we want to be
get
on topic or
doxxed.
not on topic at
I
all?
think we can be on topic. Or we can talk about whatever. You know, like, wow, we're stuck at home. What do you guys do in your free time? Something like that.
When's the last time you went outside?
Last time I went outside, like 2 weeks ago, for like an optometrist appointment. Get
Didn't
you.
you go
I didn't
outside to
even
like
know they
shovel
were
the
open.
driveway?
I don't think that counts.
Oh, okay.
If that counts, I went outside like a week ago, or like a couple days ago, shovel the driveway.
But we'll count it.
We'll count it.
A couple days.
Yeah.
Good.
Okay.
Cool.
Went outside last Saturday for work. I'm still working. Yay.
Oh my God, you're an essential worker.
Very essential.
Essential.
I need to teach kids karate, man.
Wow.
They gotta be active.
need
You're
karate.
teaching kids karate and you're teaching people biology. What a
Wow.
a paragon of virtue this man is.
Holy. Oh my goodness. Okay.
Oh man.
Yeah,
Um,
my mom did literal window shopping. Like we went out, she looked through the window and pointed to what she wanted.
You.
It was, oh man,
Did you guys buy anything?
she bought something.
Oh. Okay, well that's not exactly window. No, it is. No, yeah, it is. That's
It's just like through the window. I'm pretty sure window shopping is just like you look at stuff, but you know.
Yeah.
yeah. Yeah, window shopping is sort of like you don't buy anything, you just go to look. You know, it's what I do every time. Like, you go to like a shop where the things are so expensive, you just can't bring yourself to
That's
buy them,
true.
even if they're
That is
nice. So you
true.
just kind of go and look, be like, you're living vicariously through the through the shelves or whatever. It's like, man, imagine if that was my shelf though. All of these
Just
things.
what's the word? Manifesting.
Manifest.
One day I'll be at the shop and I will purchase something.
Plan for the future. I guess.
Yep. Fake it till you make it. You know, if you pretend to look at the expensive stuff and really consider it, one day that will be you.
One day. I mean,
Oh my god, it's been like 11 months since we've been properly out and about.
that is kind of depressing to think about, isn't it?
Yeah.
Well, if that's depressing, we'll just think about something else. What's that something? That's right, synapses. Let's go. We're back. 5 minutes is over, and the break, our self-prescribed, self-imposed one, we'll put that on hold. So let's talk about a synapse. What are the different? Yeah, okay. So I think first of all, the pertinent thing to look at is just the overall structure. What's where in a synapse? So I mean, who wants to take that one for a spin?
Yeah, I mean, I guess generally we have the presynaptic cell, so that's where the instruction is coming from, or the action potential, whatever. That's where the information is coming from.
Pre,
And then we have
as
the
in
postsynaptic
before.
cell.
Post, as
Exactly.
in after.
Wow.
You got it.
Isn't it?
We know our prefixes.
Isn't it
probably.
wacky and wild how things are named that way for a reason? It's crazy. It's like how language works or something.
It's like they wanted us to understand, but you know, only half the time. So we'll get into the words that make absolutely no sense later. But yeah, generally we have the presynaptic and the postsynaptic cell. We have the axon terminal, so that again is like the end of the presynaptic cell. And then we have something called a synaptic vesicle. And so we know what a vesicle is. It's like this little pouch that holds stuff within the cell. It's a pretty generic term, but in this case, it's going to hold your neurotransmitters. And then on the membrane of our presynaptic cell, we have voltage-gated calcium channels. And then on the membrane of your postsynaptic cell, you have the receptor for the neurotransmitters. What else is there? There's the synaptic cleft, which is the area between the presynaptic and the postsynaptic cell. I think that's pretty much it.
Yeah, that's
General structure.
the overall general structure, I suppose. Great. All right. So I guess now the nitty gritty, I suppose, and how it all happens. So here's just a rundown of generally what happens at a synapse. So you have your action potential based on some kind of stimulus input, whatever, running through the nerve, the neuron. It's coming down. It reaches the terminal, the presynaptic axon terminal. So the arriving action potential depolarizes the synaptic knob. This wasn't, we didn't say this, but the knob is sort of like the end of the presynaptic axon. It's sort of, it's sort of shaped like a
knob.
Yeah, like a doorknob.
Like a doorknob.
Yeah.
It looks like a doorknob.
Yeah, or it's depicted as one, at least. Not the other kind of knob. That's all right. Moving on. So it gets depolarized, guys. Wow. And as a result, some calcium channels open, resulting in calcium ions entering the cytoplasm. After a brief delay, acetylcholine is released through the exocytosis of synaptic vesicles. So we said vesicles before is sort of like the overall term for a thing in a cell that contains some kind of material. In this case, acetylcholine, or in the example we're using, which is a neurotransmitter, as we've already said. It goes exocytosis, exo meaning outside, and cytosis meaning movement of a cell across a membrane. I think, don't fact check that one. Basically, it spits the stuff from the vesicle outside because it fuses to the membrane of the cell, the axon. So the acetylcholine is like floating around in the synaptic cleft, and there are acetylcholine receptors on the postsynaptic membrane. So it binds there. And that protein happens to be a sodium channel. So it binds to that, and the sodium channels open, letting sodium flood into the postsynaptic neuron, which obviously makes the polarization inside, the resting potential rather, more positive. And what happens when it becomes more positive? It hits the threshold, which triggers
the
action potential.
The action potential, and thus the action potential is now transferred from one neuron to the next one.
Oh,
but
wow.
if that acetylcholine remains bound to the acetylcholine receptor, sodium will just kind of keep flooding it infinitely, and that's obviously not what you want. You don't want your nerves to go haywire. That's how you get like muscle spasms or death.
All right.
So depolarization needs to happen. So essentially, we need to get the acetylcholine off of the receptor. And what does this? What's the name of the chemical?
It's a very long name.
Protein that breaks down acetylcholine is acetylcholinesterase. Say that 5 times fast.
Let's break it down.
All right, so acetylcholinesterase, the acetylcholine part obviously refers to the chemical acetylcholine. Ester refers to the ester linkages, and ase is the ending that implies that it is an enzyme that breaks it down. Acetylcholinesterase, an enzyme that breaks down the ester bond in acetylcholine, and this results in it coming free from the acetylcholine receptor, closing the sodium channel. And after all of that, the synaptic knob reabsorbs the choline from the synaptic cleft and uses it to synthesize new molecules of acetylcholine.
Woo.
And so the cycle is complete, ready anew for whatever weird stuff you might be doing with your brain. There are some terms that we sort of didn't go through, in that whole explanation. So now we can get into the specifics of it. We'll start off with the excitatory response. It causes depolarization of the resting membrane potential, sometimes enough to cause an action potential, sometimes not. Allows sodium coming into the neurons, sodium coming into the synapse of the neuron. Basically, it turns a neuron on. It gets excited. That's the excitatory response. So a neuron needs to be excited for it to move the signal along, and so the excitatory response excites it naturally. And so this is sort of the excitatory postsynaptic potential. So that's depolarization of the postsynaptic membrane in order to propagate that signal, in order to excite it. Naturally, if there's an excitatory response, there must also, not must, but there is, in this case, the inhibitory response, otherwise called the inhibitory postsynaptic potential. So rather than sodium ions, it's something that once the neurotransmitter is triggered, it doesn't let in sodium, it lets in chlorine. So hyperpolarization occurs. Thus, it gets more negative and farther away from the threshold potential, basically limiting its ability to become, positive. And basically, it turns the neuron off. An example of this is the GABA, otherwise known as GABA chemical. So it binds to the GABA receptors. It is gamma aminobutyric acid. And so, obviously, there are some diagrams that you can look up. Our production quality isn't high enough to actually put them up, which is code for I'm lazy. But really, you just think about the threshold as being a line and the resting potential as being below it. If there's an inhibitory response, that line moves down. If there's excitatory, it moves up. So if there's both an excitatory and inhibitory response, nothing happens because it goes back down and never reaches the threshold. If there is multiple excitatory responses, it'll reach the threshold and begin the action potential. Just cool stuff all around. Nerves are great, right? Right.
Yeah. And we're done.
With
Moving
that
on.
That's it,
section,
that's the whole unit.
no,
Goodbye.
wait.
that's
No, there's more.
Yeah, but this is something like different.
Right. Well, yeah. Well,
Another
that's
word that you can
synapses.
say
That's
5 times
synapses.
fast.
Neonicotinoids.
I'll say it
I'm not
faster.
gonna try to say
5
that
times.
fast. You might
Okay.
accidentally say a slur or something, and that's
Whoa,
not
whoa,
cool.
whoa, 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 whoa.
Hey, I
whoa.
said it's bad and it would be an accident, so we're not gonna do it. All right? You can't say whoa, whoa, whoa to me saying let's not say slurs. All right? Because that reflects poorly on you, and not me. All right. So
So what are they?
neonicotinoids are a certain chemical that does a certain thing to your synapse. So we'll see the actual effect on it, but basically, it's a type of chemical that is used to protect crops from pest species. There are disadvantages to their usage, so they've been linked to a reduction in honeybee populations. Bees, of course, are important pollinators within ecosystems. If all the bees died just instantly right now, most of
very
everything else would also probably die pretty quick. There's no plants for food.
Yeah, so what? Okay, so neonicotinoids mimic your acetylcholine, but they cannot be broken down by acetylcholinesterase. So what does that mean for our neurons?
Means
And
that
it starts with S and then it ends with tops.
starts with S and ends with tops. Right. Well, that's not necessarily the case because rather than stopping, it's permanently active. It's
Yeah, new information isn't
right.
being
Yeah.
fed.
So acetylcholinesterase breaks down acetylcholine and prevents overstimulation and blockage of acetylcholine receptors. So think of it like, I don't know, a bathroom, stalls. You go in,
The
take your
beach.
dump, and you leave, right? That's normal work. So sort of the volume of dumps going down the drains is not in excess because it's one person in the stall. They take their dump and they leave for the next
Oh my
person.
god.
But if you're
This is the best.
exposed,
Okay, you know what? Continue.
if you're exposed to
Fine.
neonicotinoids, each
Yeah.
one of them binds the receptors and cannot leave. So they are just in the stall forever, just pumping out miles and miles of turds just down the drain because acetylcholinesterase cannot break them
Oh my
down.
god.
So this leads to paralysis due to the blockage of the receptors. They've clogged
Yes.
the toilets,
Yep.
basically.
Your neonicotinoids are blocking the receptors on your receiving neuron.
They make the
so that's your
stalls
postsynaptic.
unusable.
You know, As long as people remember, as long as I remember, whatever.
Right? It's see,
Yeah,
listen.
it's
Okay,
actually
okay, okay.
effective
This is what I always say.
in that
The,
sense.
the
Yeah.
more the more wacky, the more wild your mnemonic device or memory tool is, the more likely you are to remember it. Right? Like
That's a
to
fair
this day,
point.
I still remember that a poutre is like a supporting beam in a building because I remembered it as poutre me out of my misery so I can tie a rope to this beam because it's
Oh
a
my god.
supporting beam. And it is
Yep.
so terrible, but it's forever burned into my memory as a
It's,
result.
it's so bad, but it gets the job done.
It's gets the job done
That's what.
because it's so bad.
Yeah. So drugs on brain, drugs on brain.
All right, so drugs on the brain. So, I mean, people take drugs because their life sucks. Their brain doesn't work the way they want it
Okay,
to.
you don't
You know,
you don't
they,
have to go there.
Okay.
hey man, we're setting the scene. All right,
Oh my god.
Okay.
all right. So, how do okay? How do drugs work? How do drugs work? They mimic neurotransmitters. They bind to the receptors in the place of the neurotransmitters. So they allow nerve signals to be created where there initially shouldn't be one, right? So there's no, for example, serotonin makes you feel happy. I think, yeah, it has to do with sort of that
Yeah.
that pleasure center in the brain, I guess, is what it's called. And usually it's inactive unless something good happens, or like you trying to think of a safe for work example. You ace a test, or you're eating ice cream that you really like, something of the such.
Yeah,
So
baby.
There you go.
serotonin is released as a result of that stimulus. And if there's no stimulus, there's no serotonin, so you're not getting that response. However, if you take a drug such as ecstasy, which mimics serotonin, you can sort of artificially cause your brain to think there's serotonin and think something happy or something positive is going on. So yeah,
So if you're sad, take ecstasy.
that's not what I
No,
said. Don't do that.
no,
This is
don't
Don't.
do that.
great.
How about reuptake or alter?
I don't remember. You have notes on that? I don't really.
I think this is why I wrote unfinished because we
I
didn't finish
also
this
think
part.
that's why you wrote unfinished.
Could have
Well,
just left it, but no, he had to be like, when is it?
listen, man, that's not the point.
I thought you had them.
All right. You know, talk for
You're just
like.
saying it
Listen,
like
no. Talk for 2 minutes. I'm gonna find it. I'm
Okay.
going to find it.
So Sita, what you have for lunch today?
I haven't had lunch. I had breakfast for lunch. I had some oatmeal.
You had brunch?
Sure.
Cool. Just plain oatmeal?
Wait, no, just who has plain oatmeal? No.
Mm, okay.
Honey, there's some raisins, there's some
Mhm.
almonds in there. It's great. But
Oh,
yeah.
Almonds.
How is your rice?
oh yeah, I just heard the beep. Let me go check.
We are multitasking.
Yeah, brilliant. Um, here, neurotransmitter reuptake.
Or is it reuptake of the acetylcholine?
No, there's like reuptake inhibitors and stuff, but we should have that, but for some reason we don't. I guess we're just terrible at taking notes. Wait, I remember taking this though. Reuptake. Oh. Um,
Did he just say it affects reuptake? That's it?
perhaps. All
There we
right. Well,
go.
okay. Essentially, reuptake is the sort of process. I could just ad lib this. I'm pretty sure I know it. Reuptake is the process by which neurotransmitters are sort of taken out of the receptors and put back to where they're supposed to be. That's what the acetylcholinesterase does. It helps with the reuptake of it. But obviously, this reuptake system can be changed or affected if the the thingy neurotransmitter is unable to be returned to the neuron or whatever. It's going to obviously keep inhibiting that, not inhibiting, keep eliciting that response from the nerves and the action potential. And so that's the reuptake is an important part, and if you mess with it, it can be different. But sometimes it's a good thing to mess with it. For example, if some like antidepressants work in a way such
Brought
as
him back.
to inhibit the uptake. So it's like a serotonin something reuptake inhibitor. I forget what it's called. And essentially, what happens as a result is that the reuptake is inhibited, so the serotonin stays and keeps eliciting that positive response. So this is used as sort of like a medication to help people with depression.
Yay.
Something like that. Another way that this system happens is that they, instead of preventing reuptake, they simply release more neurotransmitters. So this is essentially stimulating the calcium absorption of the axon, because more calcium coming in, more neurotransmitters are exocytosed and put into the synapse or the synaptic cleft, and it's all uphill from there. I think that
Exciting.
should be it.
Yeah, we went through all the nerve things. Now,
Yeah,
There's a little bit
I think we missed something.
difference between bees. I'm not bees. Like why humans aren't as affected. So like just one small point, but I feel like we spent a lot of time on that bee video. So
Well, if you want to find it, be my guest, because
heyo.
I'm looking and I can't find it.
the bee thing.
The bee thing.
Bee thing. Okay. Insects basically have a different composition of their acetylcholine receptors.
In other words,
Basically they're what?
They're built different.
Okay.
Okay.
Yep.
The neonicotinoids basically bind much more strongly to their receptors than they do to ours, assuming we don't have any insects watching. But neonicotinoids are significantly more toxic to insects than mammals, so it makes it a very effective pesticide. But as we said, that just has severely impacted our bee population.
Sag.
Oh, is that it? That's the...
I mean, yeah, it's just like less bees. Also decreases in bird populations because there's less insects to eat.
True.
Yes.
True.
That's basically it.
Then they die.
Is chemical weapons just something we did for fun, or do we actually need to?
Yes. Take that one out of context, please. We did chemical weapons for fun.
Yep. Definitely.
There you go. There's your clip.
There, clip it. Yeah, I think that's good. I think we'll move on, and I guess we'll come back to it if I find it. So, neurons, what do they actually do? Well, they move muscles.
Class in 5 minutes.
Yeah.
Oh, you guys have to go to that one?
Yeah, it's French.
Just.
Oh no, I have econ.
Just mute it. Just mute it. It doesn't matter. It's fine.
All right.
Oh, and econ actually, it's just a documentary. Like you literally don't have to like it. Like, but actually. Anyways, muscle movements.
I think she called on me randomly in French, so
whatever. Muscle movements and joints. Someone go look at that. Start talking.
This is a whole new can of worms.
And we're opening it.
I have 5 minutes.
All right, we're not opening it.
Thank you.
Great. I guess.
There's your teaser.
Yeah, there's the teaser.
Yeah.
It's part 1. I guess. Wasn't planning for this to be 2 parts, but that's part 1. Great. So I think overall today we looked at the nerves and synapses and how all of those things work, their structure. I don't know. I already did this whole spiel. Do I have to do it again? Not really.
That's about it.
Nerves,
See ya.
synapses.
That's about it.
Etc.
Right.
Chemical synapses, neonicotinoids. That's where we stopped.
Hey man, who cares? If you really want to learn, watch Hank Green. We'll see you soon.
What? All right.
Goodbye.
Catch you next time, where we'll look at muscle movements and joints and how the nervous system interacts with all of those to make you move. I guess
my
that's our
God.
teaser. Part 2 coming soon-ish. I don't know. We studied for like an hour, and if you listen to this, so did
This is
you.
coming out.
Good
so-
job.
They know the editor to cut the clip.
Cut the clip. There's no editor. This whole thing is going unedited.
Oh,
Okay.
Yeah, I mean, sure. Hey, do your best moist critical impression, Justin. Let's hear it.
I already did it,
Do it
but
again.
you kept on talking.
Do
Do it again.
That's about it. See ya.
All right, my turn. That's about it. See ya.